All right, this is the first video in chapter 8, which is titled Protein Synthesis. And before we get too far into this, we want to review the components of DNA that we've talked about before now. So this is all going to be a review, save for the fact we want to talk about this gal and these guys. Very famous biologists. This is James Watson and Francis Crick, and I'm not sure who is who. And these guys are attributed with discovering the structure of DNA, which was super important, and they got the Nobel Prize for it. This gal's name was Rosalind Franklin, and she took the picture of the first strand of DNA, wasn't quite sure what she was looking at, but Watson and Crick used her picture to kind of deciphered it. So just as kind of a case study, it's interesting to note that Rosalind got zero credit when it came to the Nobel Prize for various reasons. But one of them was that she was a woman. And back in the 50s, women really were not only not encouraged to do science, they were looked down upon if they did. So ladies, you should be thankful that we are where we are today. It was because of ladies like her that kind of paved the way for us. So we have been saying again and again and again that DNA or genes, so remember a gene is a section, are the plans to build proteins. And what we're going to do in this chapter is we're going to look at how do we finally, we're going to look at finally, how do we put these two together? How does the DNA build a protein? Well, before we do that, let's make sure we go back over. We've got a duplicated chromosome here. So we've got our centromere. And these are going to be chromatids. We said that one section of a chromosome, so if we take a section, is a gene. And a gene is the plan to build a protein. And we're talking about a specific protein. We have alleles. So let me just color these guys in. Let's say these are two genes, two copies of genes. So they come in pairs called alleles. Also, we know that chromosomes are loose DNA that's been wound up. And so what we see here is we see it coiling and curling. When it's loose, it's called chromatin. And when it's wound up, it's called a chromosome. In order to curl up, it curls up around proteins called histones. And so what we see here are histones. And as we're going in at a more magnified view, we begin to see the shape of DNA. And we've got this unique double helix. So it twists together in a double helix. Again, this is all review, so I'm not spending a lot of time discussing it. I just want to refresh your memory. And then finally, we get down to the monomer level. The monomer of DNA are nucleotides. And remember the name for the group. So we had carbs, proteins, lipids. The name for the group that DNA belongs to are nucleic acids. So all DNA are considered to be nucleic acids. And the monomer are nucleotides. We have adenine, cytosine, guanine, and thymine. Remember the CG. And we have a complementary pairing where adenine will only pair with thymine and guanine will only pair with cytosine. So we use the whole, the monomer CG to show that cytosine and guanine will pair, and adenine and thymine will pair. One other thing to point out too, we said the bonds between these guys along the outside ridges, and this is, we've unwrapped it here, so we've taken it from the double helix into a ladder strand. This is tied together by covalent bonds. Covalent bonds. But the nucleotides here at the X's, so here's where the thymine and the adenine combine and the cytosine and the guanine combine. These are tied together by hydrogen bonds. Hydrogen bonds are weaker and so they can break. And what we're going to see in this chapter is that we need to break all of these bonds between the nucleotides, which is known as unzipping the DNA. So let's recap. You have a lot of definitions here. So we talked a little bit that was new about Watson and Crick and Rosalind Franklin, but let's check out all the definitions that you should know going into this chapter. We have chromatids, centromere, chromosome, chromatin, alleles, histones, nucleotides, nucleic acids, and then we need to know the base pairing and we need to know the bonding. So that's like 10 pieces of information that you should be able to pull back in from other chapters. Okay, so now that we have that reviewed, we can actually begin to jump into how this DNA lives in its world and how it actually builds a protein. Okay, on to the next video. Thanks for watching.